The world population as of 2014 stands at around 7 billion people and is predicted by the United Nations to increase by to 9.6 billion people by the year 2050, an increase of almost 8 billion people in just a century. Accompanying the surge in world population is the ever-increasing demand for food, a demand that the world's agricultural sector will have to adapt to if humanity is to survive and prosper. In order to feed the world's increasing population, it is predicted that the agricultural sector will have to increase yields by a minimum of 50% by the year 2050. Additionally, the impact of climate change will put further pressures on the world's agricultural sector, as formerly arable land becomes less suitable for farming and arid areas expand. Therefore, as well as increasing yields, what can we do to ensure that plants cope better in conditions that are seen less than suitable for farming?
The majority of plants on Earth use a form of photosynthesis known as C3 photosynthesis, in which the carbon dioxide is put directly into the Calvin cycle and is initially incorporated into a 3-carbon compound. In C3 plants, photosynthesis occurs throughout the leaf and is not restricted to certain areas, as it is in other forms of photosynthesis. In order to acquire the carbon dioxide necessary for photosynthesis, the plants open their stomata during the day to allow carbon dioxide to diffuse in. This form of photosynthesis evolved at a time when atmospheric carbon dioxide levels were very high and oxygen levels were low. As a result, the plants acquired far more carbon dioxide than oxygen, and consequently their photosynthetic activity was very high. Additionally, the lack of oxygen taken up, due to its low atmospheric concentration, limited competition from photorespiration. Thus, plants lost less energy and carbon dioxide to this process. However, it is the origin of C3 plants that is now a hindrance for them. The current climate is radically different from the one in which these plants evolved, with CO2 levels now very low and O2 levels far higher. As a result, the photosynthetic rates of C3 plants are far lower today than they originally were. Additionally, due to the increased atmospheric concentration of oxygen and the subsequent increase in the amount taken up, CO2 faces greater competition for the active site of enzyme rubisco, and as a result, rates of photorespiration are significantly higher. These inefficiencies restrict the amount of growth plants can undergo in a year, thereby limiting agricultural yields. Over the course of evolution, some plants evolved a new form of photosynthesis better suited for hot and arid environments. This photosynthesis is termed C4 because the first synthesized product is a 4C compound. This form of photosynthesis is used by around 7,600 species globally, including important agricultural crops such as sugarcane and maize. In the mesophyll cells of C4 plants, the enzyme PEP carboxylase adds carbon dioxide to PEP to form a 4C compound. This compound then diffuses into the bundle sheath cells, which are cells deeper within the leaf. And this is where carbon dioxide is released from the compound so that it can be fixed by the enzyme rubisco. Therefore, C4 plants have a specialized anatomy called Cran's anatomy. This enables spatial separation of the processes in which atmospheric carbon dioxide is fixed, and the fixation of the carbon dioxide released from the 4-carbon acid. This anatomical adaptation reduces the amount of CO2 lost to the atmosphere via leakage, and serves to concentrate carbon dioxide around rubisco, consequently increasing photosynthetic rates. Additionally, concentration of the carbon dioxide reduces the amount of energy and carbon dioxide lost to photorespiration, as rubisco is more likely to interact with the carbon dioxide than oxygen, as the concentration in the bundle sheath cells is significantly higher. Because C4 plants use the CO2 they capture more efficiently, they do not need to take up as much, and thus they are not required to keep their stomata open for as long as C3 plants. This is especially useful in hot, arid environments, where transpiration rates through the stomata are high, and therefore plants with their stomata open lose a significant amount of water. As a result, C4 crops grow better in arid areas than C3 plants. Therefore, GM modification of C3 to C4 plants would enable a wider variety of crops in areas that currently struggle agriculturally due to environmental conditions that limit growth, such as areas of Africa. But GM conversion of C4 crops would also be useful in areas of the developed world that are now experiencing drought as a result of climate change, such as areas of the United States and Southern Europe.